I couldn't have wished for a better introduction on Rett syndrome than what you gave here. So I was asked today to discuss a little bit on the research which I'm doing on this syndrome. I know it is a lot of hardcore science, so please don't feel frustrated if you do not understand everything I will show you, but Katrin insisted that I would speak a little bit on Rett syndrome and what we're doing. So I'm a clinical geneticist from Leuven, from Belgium. So I see all kinds of genetic diseases, but my heart is with neurodevelopmental disorders and even more with all the disorders which are associated with this special protein, which is called MECP2. There are two diseases which are associated with this protein. So the one is Rett syndrome. You see it on your left. This is the story of Franca. So there you have, women have two X chromosomes, but if you have a mutation in one of the two genes of the MECP2 gene, you have Rett syndrome, which is mostly seen in girls, and boys are not even born with the mutation, so it's lethal in boys. And on the other hand, and that's the reason why I entered the world of MECP2, is the syndrome that we discovered in 2005 in Leuven, which is a syndrome in boys where you have too much of the protein. So girls who have not enough protein, they have Rett syndrome. Boys who have too much of the protein, they have MECP2 duplication syndrome, which is also a very severe disorder with an early onset neurodevelopmental delay, severe mental retardation, and severe epilepsy. Let's see how this works. So the reason I'm talking a little bit on research is because before you can even develop therapies, we need to understand. We need to understand what the protein is doing in the brain. And it's only when you start understanding the disease that you can start trying with all kinds of drugs or molecules or even gene therapy to see if we can treat the disorder. So as you can see, and which was already thought about, is the DABU, which is the medicine which is available for certain Rett patients in Estonia. To be honest, in Belgium, we do not have access to DABU yet. And then for the moment, there are two clinical trials ongoing, mainly in the United States, which are based on gene therapy, where they try to replace the gene by adding the correct gene into a viral vector and then send it to the brain. But I'm not going to discuss on that. I'm going to discuss a little bit on what we are doing in Leuven for research on Rett syndrome. So this is just showing you the two gene therapies where they put the correct gene into the virus and then they send it or they inject it into the cerebrospinal fluid or directly in the ventricles of the brain. So what I would like to discuss with you is a little bit on what is this protein doing. And it is a master protein. I always compare it with the CEO. So it's the general manager of a huge company. MECP2 protein has a lot of functions. And I really pity my PhD students who have to write their thesis and make an introduction on this gene because it has so much functions in the brain. So MECP2 is a protein which is, I don't know whether there is a pointer, which is in the nucleus of all cells. So it has a function in all the cells of our body, but apparently the symptoms are most pronounced in the brain. So it is present in all types of brain cells, so in the neurons, as we call the major brain cells, but also in supporting cells, which are the glia cells. And it has a lot of functions. So it's really a CEO. It has a lot of management functions. It is present. It is important for making connections between neurons, which we call the synapses. It is important that we have these little dendrites that grow out of the neurons that make contact. but it's also important in the energy metabolism of the neurons, and also and mainly known because it regulates a lot of other genes. So MECP2 will decide whether other genes are either switched on or switched off. So this is what we call a transcription factor. So it has a lot of functions in the brain. And the way we have been studying the protein is by going in the lab and using different models. And the most used model to study diseases is the mouse. So there are already since the 1990s several mouse models. What they have done is that they created a mutation in the gene, and they have developed several mouse models, mice that show also some symptoms which are a bit similar to what we see in the girls. And of course, we can use the mice to test drugs and to see whether there are drugs that have a good effect and can diminish or make disappear some symptoms. What we have been using in Leuven is a complete other model, and I don't know whether you're acquainted with this one, but this is using human cells. So what we do is we make human brain cells in a dish. So we make them in the lab and we can study them. And we can even go further, we can even make small mini brains in the lab using cells coming from patients. I will give you some more information. This is a very busy slide, but basically what we do is you take some cells from a patient, and the easiest way to do this is take some skin, so little bits of skin cells, you take a small biopsy, and then the skin biopsy or the skin cells, you can add certain molecules and make these skin cells believe that they have to become stem cells again. So we make stem cells that have the same mutation as the patient from whom you took the skin biopsy. And then these stem cells here below, you can push them forward. You can say either we can make neurons, brain cells, we can make skin cells, we can make liver cells. So there are a lot of protocols that are already available to make neurons, for example. And as you can see at the right panel, so this is how these stem cells look like. This is the gray photo. And then you can push or you can add some molecules, and so that these stem cells start to become neurons. First, there are very young neurons, and then they will become bigger and bigger. And as you can appreciate, after 60 days, you see that you can really make nice neurons in your little plastic Petri dish, thank you, your Petri dish, and then you can study the neurons, and you can compare them between normal, healthy neurons with a healthy MECP2 and the neurons who have a mutation in MECP2. So this we did, we already, and this has been published already. However, we went one step further, because what is the problem? Well, these neurons, they are in this Petri dish, but after 60 days, they die, because they don't like to grow on plastic. They need blood, they need other cells, they need the environment of the brain. So what we did, and this is the work, it's almost 6 years of work of a PhD student. So what we did is we made neurons in the dish, very young neurons, and then we injected the human neurons in the mouse brain. It's really a little, well, it's millions of human neurons, but it's billions of mouse neurons. So it's not that the mouse starts speaking or thinking for itself. It's just a little amount of human neurons. But what happens is that these human neurons, if you inject them, they start spreading and they start to integrate in the mouse brain. So here, I don't know whether you see it, but what we do is the human neurons have a green tag. So they are green if you look under the microscope. So this is, for example, a mouse that was killed, after 14 days after the transplantation, you slice the brain, you look under the microscope, and see how nicely you see the human green neurons that start going into the brain. And so, of course, the longer the mice live, the more neurons will have been integrated in the mouse cortex. And this is, I don't know whether I can activate somehow the video, or do I have to push somewhere? So you see here the complete mouse brain, and you see one side how the human neurons are after, this is 7 months, how they have integrated, and they survived. They survived for a long time, and that's what we want, because now we can really have a look at how these neurons behave. And so what we did is we had stem cells, or we started with skin cells, and then we made stem cells from a boy who has the MECP2 duplication syndrome, so who has 2 copies of MECP2. Then with CRISPR, I don't know whether you're familiar with CRISPR, but you knock out 1 copy, so you cut in 1 copy. So then we had a second line where you have a normal amount of MECP2. This is our control line. And then if you cut both copies, there is no protein left. That's the Rett syndrome line. So now we could compare the 3 lines. So we have either too much MECP2, we have a normal amount MECP2, or we have no MECP2. And so we can use these lines to study both the Rett syndrome and the MECP2 duplication syndrome. And so what we've done, and I know this is really hardcore science, but I just go over it. So what you can do is, first of all, you can look at the neurons. Do we see differences between the neurons if they do not have sufficient amount of MECP2? We can look whether the neurons are functional. Do they communicate with each other? And then we can also look at transcriptomics, so this is a difficult word to say. We can look whether some genes are, as I said in the beginning, MECP2 is a switch. It will switch on genes, it will switch off genes. And so we can see whether some genes are switched on in Rett syndrome, switched off in the control, etc. So I'm not going into detail on results, but just to show you, if you want to look at morphology, as I told you, we can have a look how the neurons look like. We can even look at how well they are maturing, and maturation means that you see that the spines, and these are the places where the neurons communicate with each other, when there is interaction. If there are more and more spines, means that these neurons are becoming mature and more mature. And also, we can also have a look at how the spines look. Are they still very thin, or do you see already mature spines? So this gives an indication how well these neurons are developing after certain months. Just to show you how does it look in real life. So again, we have our 3 different diseases. So the control is the orange one. MECP2 duplication is the green one, and Rett syndrome neurons is the blue one. And you have to believe me, because I see it's very small here, but beneath you see that the control neurons, the orange, you see more and more spines, and they're more and more mature, while the Rett syndrome and the MECP2 duplication neurons, they lag behind, they're not maturing as they should. This is just giving you a... We can also look whether these neurons are functional, because what do neurons do? They talk to each other. They give electric stimulus from one to the other via the synapses, and we can measure this. So what we do is we kill the mice, I'm sorry, but you make slices, and immediately you can, with electrodes, you can measure current. So you can measure whether these neurons do communicate with each other. And if this is done in a normal way, I'm not going to show you this in detail, but maybe here you can see, I know it's quite small, but you see that, again, the orange ones are the controls, and you see that compared to the green, which is Rett syndrome, And the blue is Rett syndrome, and the green is the duplication syndrome. That, again, these neurons are less functional, there is less current, there is less communication. And then for really the diehards, we can also look at transcriptomics, which means that we look at genes which are switched on, genes which are switched off. This is really, I'm not going to go in detail because it's really quite difficult to explain, but also there we can see that in Rett syndrome, several genes are switched on, which are normally not switched on, or switched off, which are normally not switched off, and vice versa. In the duplication syndrome, we have found hundreds of genes that have been what we call differentially regulated, but we believe this is still the tip of the iceberg. And then you can even go further. You can look which genes are switched on, what do they exactly do, which genes are switched off, what's their function, and then you can establish what we call gene ontologies, but I'm not going into that. So I just wanted to give you a flavor about basic research because this is what we need. We need to know the pathways underlying the disease. We need to know which pathway is switched off, which is switched on, because then, only and then, we can start looking for new therapies, new molecules that indeed might affect the results that we see, because that's the purpose. This model we can use to test drugs, to see are there drugs that can correct the abnormalities that we have seen in our model. I know this is 6 years of work, and this is teamwork. My student is somewhere, she's the one with the orange shawl who's sitting in the front, and she will have her PhD defense this year. And thank you for listening. I know I'm the last obstacle between you and the coffee, but I'm happy to take any questions.
Yes, thank you. Kas meil on, alustame saalist, küsimusi kellelgi?
Are there any questions from the audience? Yes, please.
I can ask in English as well.
Please,
it was fascinating talk.
Thank you.
I was just wondering whether when you find new molecules that would work, can you also use your model to test these molecules in vitro rather than doing mouse studies or animal studies or even human studies?
So this is what I explained. So we also have the in vitro model where you keep the neurons in the dish. The problem is that, to be honest, after 60 days, these neurons are still very young and they do not really represent what is going on in the brain. And the big difficulty, especially with Rett syndrome, is that you have this apparent normal development the first months, years, and then you have this regression, and it's very hard to get to that phase. So that's the reason why we prefer the in vivo model, because there and even there we're not sure, because our brain takes 9 months, at least 9 months before we are born, our brain is already developing. And then you see, we believe that after 3 to 6 months, we're not sure, but after 6 months, that the neurons we are looking at in the mouse are the equivalent of newborn neurons, of a baby. That's the problem, because our brain is already maturing in utero, and so therefore I think all the in vitro studies have more limitations.
Saalist veel küsimus, jah.
Thank you for the nice talk. So my question is about dosage. So MECP2 is a dosage-sensitive gene. Too much is bad, too little is bad. So now I know that there are also drug developments, gene therapy, also downregulation with antisense oligos. So how to actually make sure that you hit the correct dose? That when you try to upregulate this gene or add this gene that you don't add too much, and vice versa?
That's a very, very good question, and you have clearly understood the message, which is already very important. Yes, so that's of course what a bit of the clinical trials is about. I can talk about the ASO therapy for the MECP2 duplication, where we try to downsize the amount of MECP2 protein. So they're titrating to see whether, but it's very hard but we have quite a large window. With regard to Rett syndrome, where we have to increase the amount of the protein, there the two gene therapies which are currently underway, they have a kind of a sensing molecule that senses the amount of protein which is made, so not overshoot. But again, they start with low dose, and that's the whole thing of the clinical trials. And then, of course, higher doses, you have the disadvantage of the AAV vector, etc. But this is something that is absolutely taken into account, and I think it's a bit of a trial and error almost. Yeah. I think we do our best in the beginning to be very cautious, but it's very hard because we do not have very good biomarkers. At least I'm not aware of good biomarkers.
Saalist küsimusi? One question.
Other questions from the audience?
Viewers. What do you think when we have a very short time from first symptoms to diagnosis, can we prevent some or most of the symptoms of Rett syndrome using debut or some other treatment?
Well, that's a difficult question. It's of course, the earlier you treat, the better. I think this is true for all rare diseases. And so it starts with early diagnosis, and then we come into the discussion about newborn screening, because there is also a lot to do about, shall we start by doing a genome analysis in every newborn? I know also in Belgium there are big discussions to have a very, very early diagnosis. But of course, if you have an early diagnosis, you also should have something to treat immediately. So both have to be there. But of course, I think the general message is the earlier you treat, the better the outcome. Well, that's my opinion, but maybe somebody else has another opinion. But
how much can we hope that the gene therapy will help Rett syndrome patients in the near future?
Well, I'm hoping with you, because I don't have not a lot of results yet, but I think we're waiting for the first results of the trials, which hopefully come already this year. We have some first results.
Saalist küsimusi? So thank you, Tartu Ülikooli Kliinikumi laste neuroloog, Leuveni ülikooli haigla tippspetsialist ja Tartu Ülikooli.
Thank you to the top specialist from Leuven University, Hilde van Esch. Thank you.